So hello guys, welcome to my YouTube channel Med School with Shubham. I am Shubham Mane, a third professional MBBS student, and I shall be teaching you anatomy. I have started a new series on embryology on my YouTube channel in which I have completed the first week of development and the second week of development, and I also started the third week of development. This is the part 2 of the third week of development, in which we will be learning about the formation of the notochord. In my previous lecture, that is part 1, we have studied about the process of gastrulation, in which I taught you about the primitive streak, primitive node and the prochordal plate. So this is the extension of the previous lecture, in which we will be learning about the formation of the notochord. So what actually is notochord? So notochord is a midline structure. Notochord is a midline structure which develops in the third week of development. It is a midline structure. It develops in the third week of development. It develops from the primitive node. It develops from the primitive node and it extends from the primitive node to the prochordal plate. I hope this is very clear to you guys. So what's the definition of notochord? So notochord is a midline structure in the embryo which develops in the third week of development and which develops from the primitive node and extends from the primitive node to the prochordal plate. So let me show you this in the diagrammatic form. I hope you remember this diagram, guys. Guys, if you have not watched my previous lectures, I request you please go on my YouTube channel and watch my previous lectures on embryology. So let's continue with the lecture. I hope you remember this. I have taught you in my previous lecture. This is the embryonic disc or embryonic. This is the embryonic disc. This is the prochordal plate or the prechordal plate, and this is the primitive streak, and this is the primitive node. The primitive node is also called as the Hansen's node. Now, according to the definition, I said that it develops from the primitive node and it extends from primitive node to the prochordal plate. Means what? It will start from the primitive node and then it will run towards the prochordal plate. Means it extends from the primitive node and it will run towards the prochordal plate till the prochordal plate. So a new structure will be formed in this region, which is called as the notochord. So now we will be learning about the formation of the notochord. So let's get started with the formation. Guys, in my previous lecture, I taught you about the process of gastrulation. So here the gastrulation has taken place completely. So now we have the definitive ectoderm, the intraembryonic mesoderm and the definitive endoderm. So accordingly I will be making the diagrams. This is the definitive ectoderm. Then we have this primitive node. This is the primitive streak and this is the definitive ectoderm. The same thing. I hope you remember this thing. Then beneath that we have this intraembryonic mesoderm. And beneath that we have this definitive endoderm. So let me name all these structures. Okay. So this is the ectoderm, this is the endoderm and this is the mesoderm. Okay, and this is the primitive node, and this is the primitive streak, the primitive node and the primitive streak. Okay, now let's move forward, guys. I hope this is very clear till this point. So let's move forward now. Now what will happen? This primitive pit will move more deeper inside, and now we can see a structure like this. This is the ectoderm. Okay. Then we have this primitive node, and now the pit has gone more in deeper. This is the primitive streak. Now what will happen? This is the intraembryonic mesoderm. Okay. Now the next thing that we can see is inside this primitive node, some specialized cells will be formed. Inside the primitive node, some specialized cells will be formed, and they will migrate beneath this intraembryonic mesoderm. It will go inside the intraembryonic mesoderm. So it will form a structure which is like this. They will form in a mass-like structure in this manner. Okay, and this structure is called as the notochordal process. Notochordal process. Let me take a bit. Okay. Now let's move forward. Now what will happen? This pit will move over more inside. This is the endoderm. Okay. Again we have this ectoderm, then the primitive node, similarly, and now here we will be having the notochordal process, which has now extended more towards the prochordal plate. Assume that here we have the prochordal plate, somewhere here we have the prochordal plate, and this primitive this notochord will be extending from the primitive streak to the prochordal plate. Okay, so now it's extending even more deeper. Okay, now let's name these structures once again. So this is again the same thing, that is notochordal process, but now this pit is no more a pit. Now it is called as the notochordal canal, because it has become a canal now. So it is called as notochordal canal, and it is surrounded by the this is surrounded by the intraembryonic mesoderm, and below that we have this endoderm. Okay, now what will happen further? So now before that I will just cut this diagram from this side. Okay, and I'll make it over here. So what we can see is, now I have cut the diagram in this direction. In this direction I have cut the diagram, and now I will watch it from here. Okay, so I will see something like this is the ectoderm, below that we have the endoderm, in between we have the mesoderm, and here we have this notochordal process. I hope this is clear, guys. This is the ectoderm, this is the endoderm, this is the mesoderm, and this structure that you can see is the same thing that I have drawn over here. Okay, and this is the notochordal process. Okay, now again I will cut the same thing in this direction. I mean in this diagram also I will cut over here in this way. Okay, and I will drawing it over here. So it will also appear a bit similar to that. This is the ectoderm. Then you have this endoderm. In middle you have this mesoderm. And in the mesoderm you have this notochordal process, but in the center of this notochordal process you have this opening which is called as the notochordal canal. So I will be drawing this notochordal canal also in middle, and this is surrounded by the mesoderm. Okay, so let's move forward now. Now what will happen in the next step, I'll show over here. This notochordal process, it will now start moving downward. It will start moving downward. It will move in the downward direction, and slowly this part of the notochordal process, it will fuse with the endoderm. I hope you understand. It will first move downward. So now you can see over here like this, and now as it is moving downward, when it reaches over here in this way, so this part it fuses with this endoderm, and this part it fuses with this endoderm, and now a opening can be seen in this part. So let me draw that once again. So now it will be seen here. So this is the endoderm. Then we have this. Okay, and now in this part we have the okay, and in the surrounding we have this mesoderm. So let's name them. Now what is this structure form? See, this is an opening here. Okay, this is an opening. What happened here? This notochordal process, this notochordal process, it migrated in the downward direction, and then the ends of the notochordal process, it fused with this endoderm, and then it formed a structure in this way. It moved downward, and then the ends fused with the endoderm, and then it formed an opening over there. So this structure which I have made here, it is called as the neuroenteric canal. This thing is the neuroenteric canal. Now let me draw in this format also. How will it look in this format? So this is the ectoderm. This is the primitive node, and this is the primitive streak. It moved in this way. Okay, so now it had started moving downward. It had started moving a bit downward, and then this also fuses over here. I hope this is very clear, guys. And now this is called as the neuroenteric canal, and this neuroenteric canal forms a direct connection between the yolk sac and the amniotic cavity, because this is the ectoderm. Earlier it was epiblast, and epiblast was surrounded in the upward direction. It had the new amniotic cavity, and this was the endoderm. Earlier it was the hypoblast, and below the hypoblast we had the primary yolk sac. So now a direct connection is formed between this thing, the amniotic cavity and the primary yolk sac, and this is called as a neuroenteric canal. This is the same neuroenteric canal. Okay, and here you have this intraembryonic mesoderm. I hope this is very clear to you guys. So now let's move even more forward. Now what happens in the next step? So this this thing, that is the notochordal process. This is the notochordal process, and this is the notochordal canal, but now it is called as the neuroenteric canal because it is forming a direct connection between the primary yolk sac and the amniotic cavity. So now this neuroenteric canal will now dip more deeper. It will go more deeper like this, and it will form a deep structure, a flat like structure over there. It will go more deeper. It will go, first it was like this, now it will become more deeper. It become a flat like structure. First it was rounded, now it become flat like structure, and now that is called as notochordal plate. So here we will get the formation of the notochordal plate, somewhat like this. This is the notochordal plate. See the difference between the notochordal plate and the neuroenteric canal. See, here it is more often like a flat like structure. This is a flat like structure, therefore it is called as a plate. And here it is not flat, here it is rounded, therefore it is called neuroenteric canal. And this is the notochordal plate. So let me name that notochordal plate. Let me make in this diagram also. The ectoderm, beside that we have the endoderm, okay. Let me make over here downward. Now in this diagram you can very clearly see that it is very close to this opening, therefore it is forming a roof like structure or a plate like structure, therefore it is called as the notochordal plate. And this is the intraembryonic mesoderm. Okay, now what will happen in the next step? This notochordal these ends of the notochordal plate, it will get detached from this part, from the endoderm. The ends of the notochordal plate, this is the notochordal plate and these are the ends of the notochordal plate. So the ends of the notochordal plate will get detached from the endoderm. This is the endoderm, these are the ends of the notochordal plate. So it will get detached from the endoderm, and then it will form a mass like structure, a solid mass like structure will be formed longitudinally in this manner. This is a solid mass like structure longitudinally, and that is called as a definitive notochord. So let's make the diagram for that now. This is the ectoderm, this is the endoderm, in the middle you have the mesoderm or the intraembryonic mesoderm, and now these two ends have been removed. These two ends, this one and this one, these two ends are now separated from the endoderm, and now they form a solid mass like structure over here. This is called as the definitive endoderm, definitive notochord. I hope this is very clear to you guys. First we had the formation, let me make here also in this diagram, then we'll revise once again everything. This is the ectoderm. This is the endoderm. In middle you have the mesoderm. This is the notochord. Proper notochord is there. Definitive notochord is formed now from the primitive string, and here you have this precaudal plate. According to the definition, I have drawn the same diagram, guys. Okay. Let me name that now. This is the definitive notochord. Notochord. Okay. So let's revise everything once again before moving forward. So first we had the formation. So this is the ectoderm, this is the endoderm, and this part is the mesoderm. This is the primitive node and this is the primitive streak. And somewhere here we have the procaudal plate or the precaudal plate. Now this is the primitive node. Inside the primitive node we get a depression which is called as primitive pit. This is a depression which is called as primitive pit. Now after the formation of primitive pit, we will get the formation of the notochordal process from the primitive node. From the primitive node, some cells will form a solidified mass and then they will invade inside the mesoderm, and that is called as the notochordal process. Now this pit will also move inside, grow inside that notochordal process, and now that pit is called as notochordal canal. So notochordal canal will be present within the notochordal process. So this is the notochordal process, and in the center you have the notochordal canal. Next, this notochordal canal and the notochordal process will grow downward and forward. Downward and forward means it will grow slantly, and then it will go and fuse with the endoderm. It will grow downward and forward, and it will fuse with this endoderm. It will fuse with the endoderm, and a direct connection will be formed between the amniotic cavity and the primary yolk sac. A direct connection will be formed between the amniotic cavity and the primary yolk sac, and this is called as a neuroenteric canal. This is called as neuroenteric canal. The next thing is now this rounded neuroenteric canal now becomes flattened to form notochordal plate. Now it forms the notochordal plate because it is more or less a flat like thing for forms of plate. Therefore it is called as notochordal plate. And now the ends of the notochordal plate, these are the ends of the notochordal plate. This is the notochordal plate. These are the ends of the notochordal plate, and this is the endoderm. So the ends of the notochordal plate will get detached from the endoderm, and this notochordal plate will now fuse together to form a rod-like solid mass, which is called as a definitive notochord. See, in this manner it has formed. This is called as a definitive notochord, and it will and this is the final structure of the definitive notochord. It will grow from the primitive node, and it will extend till the procaudal or the precaudal plate. I hope this is very clear, guys. This is in the form of a flowchart that I have written. On the day 16, the proliferation of primitive node will take place. The primitive pit will be formed. Due to the proliferation of the primitive node, the notochordal process will form. After the formation of notochordal process, we will get the notochordal canal. Means this pit will grow inside the process to form the notochordal canal. Then it will further grow forward to form neuroenteric canal. Then it will form the notochordal plate, and finally it will the ends of the notochordal plate will get detached from the endoderm, and they will fuse together to form a solid mass, cylindrical mass like structure, which is called as a definitive notochord. Now the last thing which we have to learn is the fate of the notochord. So in humans, the notochord gets disappeared or degenerated. Or disappears. appears. But the remnants are still present inside the humans. The remnants of the notochord are still present, and it's very important to know the names of those remnants. So the first remnant of the notochord is nucleus pulposus. Pulposus. Nucleus pulposus. Then we have the pulposus. Then we have the apical ligament. Apical ligament of dens. Apical ligament of dens. And the third thing we have is the membrana tectoria. Membrana tectoria. The first remnant of the notochord is the nucleus pulposus. The second is the apical ligament of dens, and the third is the membrana tectoria. I hope this is very clear to you guys. This was all about the notochord, the formation of the notochord and the fate of the notochord. So this marks the end of this video, that is part 2 of the third week of development. In the next lecture, we will be learning about the process of neurulation or the neural tube formation. That is also a very important lecture in the third week of development. So this marks the end of today's lecture, guys. Thank you for being with me till this point. Please like my video, share my video, and also subscribe to my YouTube channel. If you have any doubts regarding the formation of the notochord or any other doubt regarding any other thing, you can directly get connected to me on my Instagram handle. I have shared the link of my Instagram page in the description. You can DM me directly regarding any doubts, or you can also ask me in the comment section. I hope you like the videos. If you have any suggestions regarding the lecture, are you comfortable with the language, or are you comfortable with my speed? If you have any suggestion regarding that, please let me know. How can I improve in making the videos? Thank you so much, guys, and please like my video.